The objectives of this presentation argues that women matures psychologically faster than men. The evidences that will be taken into account and evaluated is the sex dimorphism in the rate of brain development, in which scientists have showed results from MRI scans that some parts of the female brain actually develops at a faster speed than in males, which actually impacts on the maturity rate, thus providing us with evidence of psychological maturity rate. Another evidence is pubertal development, hormones, testosterone. So testosterone in this investigation contributes to male brain activity, which supports the theory, whereas women's matures faster. Lastly, circulating anti-Müllerian hormone, which is also abbreviated as AMH. This AMH is only found in men and boys, and it actually slows down the cognitive abilities in males. So firstly, evidence 1. I'll be talking about sex differences in the adolescent's brain. This research investigates the differences in the adolescent's brain. The purpose of the investigation is to better understand how brain development affects the divergence between female and male characteristics, behaviors, and etc. So using multiple sources found by other peer review associates, as well as other sources from books and theories, the research have actually shown solid evidences that many of female brain components have developed earlier than males, which actually also affect on the behavior of females. For instance, since seen in the page here, there's a comparison between the brain development of males and females. So in female brain development, it is seen that the GM volumes of the brain actually reaches peak state earlier, while for male, WM volume increases more rapidly. So the woman brain size develops at 10.5 years, and men to be 14.5 years. So actually, if you notice here, there's a 4 years of difference between when male and female actually reaches the fully development of the brain. Moreover, for brain components, corpus callosum and hippocampus actually grows larger and earlier in females, and whilst for male, the lateral ventricles is larger, and the amygdala size actually rapidly grows more than in females. And lastly, the research also identifies that there's more regional activities in male, while for female there's... Okay, so the research also explains the function of these brain regions, and here you can see, for instance, the corpus callosum, the green area, it is a bundle of nerve fibers that connects the left and right hemisphere together and helps both sides to communicate, in which previously mentioned corpus callosum is seen to be bigger in female. Moreover, explains to why female tends to have bilateral activity in their brain activation. In addition, hippocampal also tends to grow earlier in female, and hippocampal is actually responsible for learning and memory. So why is actually hippocampal relevant to this theory? So cognitive maturity requires the ability to learn and store memory for future uses and recall. Hippocampal fully develops earlier in women, actually supports women to achieve cognitive maturity earlier than men. And lastly, amygdala. The amygdala is a component of the limbic system, which contributes to the roles in behavior and emotions. The brain components is actually known for being responsible for the action of fight or flight in psychological terms, otherwise generally known as fear reaction. So, in other research, it has been shown that men tend to feel fear more and react inappropriately when sudden situations comes to them, which overall, to sum up the research, female brain volumes in the frontal lobe reaches peak growth 2 years earlier than male, which could indicate to earlier maturity rate. Thus, as both brain components, hippocampus and corpus callosum, has a larger size and growth rate, this also indicates the earlier activation of the cognitive abilities. And this research discovers how testosterone levels in healthy men are related to cognitive abilities. So when a male reaches puberty, that is when the brain, the hippocampus, it sends signals to the pituitary glands, and from there into the testes. And these testes to produce a hormone called testosterone. Moreover, only when male reaches puberty, this is when testosterone is developed rapidly. This research identifies that testosterone plays an important role in activating the amygdala function, in which testosterone could have an impact on memory function in males. The research consists of 234 healthy Caucasian young men and women, and so these young men and women is in the age range of around 18 to 32 years old. These people were asked to look at varying images, and they were put under the MRI scan as well as onto a saliva test for the testosterone. And one more thing, it's called the free recalling test. Participants' results of testosterone tests have shown very obvious result that regarding men having higher levels of this hormone. Results from the image test have also shown positive correlation between testosterone levels and the amygdala activation during encoding of neutral versus campal pictures in men. Another finding is that acute testosterone levels in women did not correlate with memory performance, that the tendency of better memory for neutral information in women as compared to men is likely to independent of sex-dependent differences in testosterone levels. So in conclusion, this findings indicates that in men, high testosterone levels relates to increase in emotional arousal, greater amygdala reactivity, and therefore to having better memory. So linking back towards the main theory, it is seen that on average, women tends to have symptoms of puberty earlier than men, of average 1 year. So it's seen in the previous page. So girl stage of puberty is around 8 to 14 years, while boys is a bit later, 9 to 15 years. This finding, although does not clearly state that men have later rate of maturity than women, but it demonstrates how the production of testosterone heavily impacts on men's learning and memory ability, which overall cognitive ability. To be able to have testosterone, to have better cognitive ability, men only produces it when they are under puberty. And we can see that men, they don't reach puberty earlier as women. Therefore, overall sums up to supporting the theory that women actually psychologically matures faster than men. And lastly, so what is AMH? AMH is a Mullerian inhibiting substances, MIS, is a tissue specific to the TGF-beta superfamily growth factor. AMH is secreted by the immature Sertoli stem cells, SC, and is responsible for the regression of the Mullerian ducts in the male's fetus as part of the sexual differentiation process. So AMH serum concentrations are elevated in males under 2 years old and then progressively decreases until puberty. So this is seen that AMH only appears in male. So this research actually uses the fMRI, longitudinal magnetic resonance imaging studies of human brain development have established that boys on average take 2 to 4 years longer than girls to reach peak brain brain development depending on the brain regions. So the results of this research actually shows that AMH may be relevant to the normal development of the human brain, which indicates that boys tend to mature at a slower rate than girls, as AMH actually slows down the process of cognitive development in male. So how this one was, how the research was conducted. The research tests maturity based on children drawings of people progress through a series of landmarks beginning with disordered scribbles and progressing to stick figures and then to elevating more complexity of drawings. So the method was that 134 boys and 38 girls at the age of around 5 to 6 years old were given instruction to draw a picture of a person. Each drawing is scored with a 23 scoring element. So the blood sample, and not only that, blood samples were taken in order to measure the AMH hormone inside the body. So each child's blood was drawn and incubated for 30 to 6 minutes to clot. Then the serum of AMH was then removed and divided, and then it was stored under 80 degrees Celsius. AMH serum was then measured in a machine called ELISA, and blood samples owner were unrevealed to experimenters. So as you can see here, there's two figures, figure 1 and figure 2. This is the results showing from the research, in which figure 1 featuring a comparison between a 3 drawings ordered in high to low levels of AMH levels and 1 drawing by a girl. So you can see boys. The boys' drawing are very simple, while the girls is more complicated. And this one actually was also measured under a maturity index scoring board. So the boys' MI score being positively associated with age in the left-hand side and negatively associated with AMH level in the right. The graph shows a trend that as the maturity index increases, the level of AMH decreases. So overall, concluding, boys have high levels of AMH and only female-like levels of testosterone. Throughout the research and comparison between boys' drawings and the girls' drawing, it is seen that there is a very big difference between maturity of boys and girls, indicating that AMH does slow down the cognitive abilities in boys. And relating this back to the key topic showcases that because of AMH, this also supports the theory that girls mature psychologically faster than boys. So source 1. And the pros of this source is that it by understanding the rate of maturity between women and men, it actually helps us identify certain psychological disorders such as depression. The research of cognitive ability in boys and girls actually suggests that sexual dimorphism in human brain, it can predict potential behaviors, which is very beneficial for things like education, where you can understand why student actually acts in a certain way. Source 2. Emerging to puberty means adolescents facing adults' emotional challenges. By knowing that, testosterone is one of the main factors that impacts on one's behavior or one's memory, it helps prevent unwanted behaviors in male, and it gives us more insights of why it's causing this problem. Source 3, the pros of this is that AMH level measurement can indicate male students' cognitive ability, which again links back to education, as it helps understand students' academic achievement during early childhood. So as you guys have early childhood, boys are more active, they do not listen in class, and etc. And we can come up with better curriculum, better learning methods for them. And the cons of the sources. So for source 1, it does not take into account a specific age group during research, as some brain regions grow at different rate, gender, and age, which could cause inaccuracy And the research collects different theories from different sources to convey the idea. So this could actually inform small misconceptions. So source 2, this source only measures boys' reaction to indicate testosterone as an indicator affecting maturity rate. So extreme maturity index can be only measured using drawings of young boys. And even in the research itself, it does state that one regulator cannot define a complex theory such as maturity, because maturity actually can be defined through a lot of factors, not just drawing. It could be reactions, it could be emotions, behaviors, and etc. Not only that, but the AMH ELISA machine is unable to separate the pro-AMH and the AMH-HNC, which could cause some inaccuracies to the research, because AMH is actually has two subcategories. So one is pro-AMH and AMH-NC, which each of these separate hormones could affect certain things in the brain. So in conclusion, through the analysis of sexual dimorphism in the human brain, hormonal changes during puberty, testosterone, and AMH level affecting maturity, it could be seen that all evidences actually shows that boys tend to mature at a slower rate than girls, therefore ending up to